11-1, we went through the history, or we talked about Gregor Mendel, who is the father of genetics, and we briefly went through some of the key vocabulary words that we're going to be using when we talk about Punnett squares. Section 11-2 is going to be on probability and working with Punnett squares. Probability is the likelihood that a particular event will occur. And it can be written as, and if we look at the diagram over here on this side, if we were to write this as a fraction, it would be 1/4, 1 out of 4 total. It could be written as a percent, 25%, or it could be written as a ratio, which is 1:3. You'll have to remember that when you're talking about a ratio, these two numbers have to add up to the total number of items. So that's why this is 1:3. Coin flipping. There are two possible outcomes if we flip a coin, heads or tails. The chance that the coin will land on 1 is going to be fraction, 1/2, percent, 50%, or ratio is 1:1. Alleles segregate randomly, just like the flip of a coin. So you can use probability to predict the outcome of genetic crosses. There are two types of genetic crosses. Monohybrid genetic crosses. Mono means one. So this is a cross involving a single trait. For example, flower color. The dihybrid cross is a cross involving two separate traits. For example, flower color and plant height. We will begin by learning about a monohybrid cross, and then we will move to the dihybrid cross. Results of monohybrid crosses are inheritable factors or genes are responsible for all heritable traits. Phenotype, and if you recall, phenotype are the physical features, is based on genotype, which is the gene combination. So the gene combination are the letters that we're putting together, and the physical features are what it looks like. Each trait is based on two genes. So once again, each trait is based on two genes. One comes from the mother, and the other comes from the father. True breeding individuals are homozygous, which means that both alleles are the same. And we talked about big R, or little r. These are homozygous, and if you have these would be, another name is pure. Punnett squares are used to show possible offspring from a cross between two parents. Parent alleles go at the top and on the left. So this is one parent and this is the other parent. So the parent alleles go on the top and down the side. The boxes show the possible offspring combinations. So the children from the parents are going to go in these boxes right here. In pea plants, tall is dominant over short. And the first thing that we have to do is assign letters when we're making a Punnett square. We are going to use capital T for tall and lowercase t for short. So let's make a cross. We're going to cross a pure tall plant with a pure short plant. What do pure tall parents look like? So what are the alleles? Remember, we used capital T for tall. If it's a pure tall, it's going to have 2 capital T's. And this is homozygous. The gametes, and what the gametes are egg and sperm. So what is going to be in each of these right here that came from the parent? Capital T and capital T. For a pure short parent, what are the parent alleles? We said that lowercase t or little t stands for short. So 2 little t's, because it's pure short, that is homozygous for that particular trait. So one gamete is going to have a little t and the other gamete is going to have a little t. So once again, when we're making our Punnett square, you need to remember that one parent goes on the top and the other parent is going down the side. These are the alleles. If we're going to fill in our Punnett square, we take this T and bring it down, and we take this T and take it over. So if we're going to fill in the next box, this T comes down, this T comes over. This T goes down, this T goes over. This T goes down, this T goes over. One thing that you need to remember is that you always put the dominant letter first. If we read the Punnett square, all of the offspring this one is tall. This one is tall. This one is tall. This one is tall. All of the offspring, or 100%, four out of 4 will be tall. Big T, big T, little t for the genotype. And they're all tall. So 4 out of 4 will be tall. Now we're going to look at what it is if we do a hybrid tall parent. So the letters that go together are going to be in a hybrid. We're going to have one dominant and one recessive trait. And these are called heterozygous. What can the gametes look like? One big T goes in one and a little t will go into the other. So if we are doing our Punnett square, the one parent goes on the top, big T, little t. Remember, we're crossing two hybrid tall parents. Big T and little t go down the side. Now, as we're filling in our Punnett square, big T, remember this one comes down, this one goes over. Here, we're going to put big T, little t. Always put the dominant one first, big T, little t, little t, little t. When we're talking about genotypes, remember that those are the letter combinations. Genotypes are the letters that we're putting together. So we've got 1 out of the 4 that's going to be big T, big T. Half of them, right? There's 2 that are going to be big T, little t. And 1/4 is going to be little t, little t. Remember that phenotype is the physical features or what it looks like. And we said big T equals tall and little t is short. Okay? So 3/4 or 75% are going to be tall. Because as we're looking, this is tall, 2 big T's. This is tall because the dominant outweighs the recessive. This is tall, so we have 1, 2, 3 tall. This one, on the other hand, 2 little t's, is short. The only way that you can have a short is to have 2 recessive alleles. So 1/4 or 25% are going to be short. And now it's time to practice and work on the monohybrid problems.